cruelty, but you've all been incredibly patient, so I'm grateful for your patience. My name is Kush Baum. I'm the president of American Society of Geriatric Otolaryngology, and I'd like to welcome you to the final 2021 webinar of the American Society of Geriatric Otolaryngology. We've had an incredible year. We've had fantastic speakers. Our year started with Justin Gallup, who gave a fantastic presentation on age-related hearing loss. And then in June, we had a presentation led by Dr. Puskis covering a perioperative care of the geriatric patient. And on that panel sat our past presidents, Dr. Eibling and Dr. Tulanay Uger. And then in September, we had Shahrukh Jalisi talk about head and neck cancer management in the geriatric patient. And now we come back full circle, back to sensory biology, sensory neurosciences, or sensory otolaryngology, if you will, with an amazing speaker. And again, I want to thank him for his patience with us. We had actually 38 registrants for this meeting. I'm sure as a result of this mishap, we've lost several. So I apologize to all of you. Dr. Pinto is our guest speaker. Dr. Pinto is professor of surgery and director of research, as well as director of otolaryngology, or rhinology at University of Chicago, Basin section of otolaryngology, head and neck surgery. He wears many hats. Not only is he an amazing clinician, educator, but he's a superb investigator who's NIH funded, and his institution has tapped into his skills as an administrator, if you will. He serves as Dean of Faculty Affairs as well. So University of Chicago is very lucky to have him. Dr. Pinto completed his medical school at Stanford University and went to University of Chicago subsequently for a fellowship. He joined the faculty at University of Chicago in 2004. His clinical practice centers on endoscopic approaches to upper airway, and he manages diseases from full range, from inflammatory diseases all the way to anterior skull-based tumors. He is an NIH-funded investigator, as I pointed out, and he focuses on risk factors for olfactory and airway disorders in a multifaceted program that involves collaborations with multiple disciplines, including epidemiology and gerontology and environmental health, sociology and genetics. So we're very pleased to have him. And so without any further ado, not to keep you guys all waiting, I'm going to turn the platform over to him. After his talk and questions are answered, I'm just going to review with you what needs to be done, so those of you who would like to receive CME can successfully accomplish that. So Dr. Pinto, the platform is yours. Thank you again for both your patience and your agreeing to be our ultimate speaker of the 2021 calendar year.
Great. Let me share my screen.
Perfect. We're able to see your screen.
Perfect. All right. Well, thank you, Kush, for this kind introduction. We've known each other for many years, and it's nice to reconnect here in the American Society of Geriatric Otolaryngology. And I guess for all of us on the call, we're very interested in older patients and how our field can take care of them better. And so I'm going to talk today about olfaction and aging. And I titled the talk, "It was the best of times, it was the worst of times," because as we've seen in the pandemic, olfactory loss is a feature of COVID-19. We see it in the news, we see it in our clinics, and there's a growing number of people, young people, who are having post-viral olfactory loss. And so this special sense that's very important is finally getting some attention. Unfortunately, it's because of this big problem that we have. So just for disclosure, I'm funded by the NIH. NIA. I sit on some advisory boards for companies that make products primarily for sinus disease, inflammatory disease, so not really related to olfaction. And I do some clinical trials for those companies. And I speak for those companies. I have no other financial conflicts, and honestly, nothing that I say today is going to be relevant for these conflicts. So the classical senses allow us to interact with others and the world around us. And I think in large part, many of us went into otolaryngology because of this, because of these primary functions of the cranial nerves, of the head and neck that really are important in communication. And, you know, this involves hearing, which we deal with, taste, which we deal with, touch in the face, which we deal with, and also the sense of smell, and that's what I'm going to focus on today. So, even in ancient times, even if as far as back as Shakespeare, the sense of smell has been recognized as being really important. Olfaction is a critical part of our physiology. It's important for human quality of life. It serves as an early warning system in terms of toxins in the environment, smoke and poisons and other noxious things in our environment. It is really involved in the sensation of pleasure. And this is everything from, being sated after meals, enjoying good food, enjoying, the smell of a newborn baby or flowers or even sexuality. It has a role in kin recognition, so mice can recognize how they're related by proteins excreted in the urine. And even in humans, my colleagues Martha McClintock and Carol Ober have shown that the sense of smell plays a recognition in potentially even in human mating choice. It's involved in pheromone detection, mood and mental health, especially regarding emotions, and really has a close connection to the brain. In fact, the olfactory nerve is directly connected to the brain, as you know, and plugs in without branching in the thalamus directly into some ancient parts of our brain, and I'll get into that in a minute. And because of this evolutionary nature, imagine even single-celled organisms need to move towards nutrients and need to move away from dangerous substances. It's evolved from this ancient role to being a really critical part of our physiology. So it really greatly affects older adults. As with the other senses, Richard Doty, recognized in a seminal paper in Science in 1984 that the sense of smell declines with age. And so this affects activities of daily living, such as detection of spoiled foods, smoke and gas leaks. Up to 65% of older adults can't detect the odor and the warning odor in natural gas, that sort of sulfurous odor. It affects eating and cooking and has been shown to be involved in nutrition. It affects personal hygiene. Imagine if you can't tell, you exercise or it's a hot day and you can't tell if you need a shower. People get very anxious about that. It affects how clean you can keep your home and affects how you buy fresh food. And then because a lot of what we do is around eating, it can affect socializing and social function. Imagine if you go out to dinner and somebody tells you, hey, there's this great new restaurant, and you really can't taste the food, most of which is mediated by smell, then it really affects how you interact with other people. And because of these things, it can really cause increased symptoms of depression. It's been associated with depressive symptoms, weight loss, appetite, and psychological well-being decline. And overall, you get decreased pleasure in life and quality of life. One thing that I think about and has been recognized for some time is that olfactory loss or sensory loss in general, this is also true for hearing, is associated with major health problems. In particular, both of these sensory losses are related to Alzheimer's disease and its precursor, mild cognitive impairment. Decreased olfaction has been found to be a marker of brain pathology at autopsy. It predicts future cognitive decline. It predates other forms of brain dysfunction or neurodegeneration, such as Parkinson's disease, by up to 4 years, and other forms of dementia, such as Lewy body dementia. And even common diseases like diabetes, renal disease, epilepsy also have some associations with olfactory loss. So these are big, important problems. Until COVID, if you had told people, well, how big a problem is olfactory loss, many people would not think it's very important. But now you see lots of people seeing the importance of the sense of smell. How does this process occur? So odorants diffuse up to the roof of the nasal cavity. There is the olfactory epithelium, which comprises the receptor surface on the superior portion of the middle turbinate, the superior turbinate, the septum, and the cribriform plate. This epithelium is bathed in a blanket of mucus produced by Bowman's glands, and those glands also produce, in this mucus, the proteins which protect and carry ASGO molecules. This is the only portion of the body where the nerve tissue is directly exposed to the environment, so consequently it's susceptible to injury, effects of things like allergens and pollutants and viruses, certainly bacteria, anything that can get into your nose can get to your olfactory nerve. And as I mentioned earlier, the olfactory nerve is directly connected to your brain, so it's a conduit right to the central nervous system. And in fact, in animal studies, you can put things in the nose and watch particles go up into the brain, and we're working on that question in human studies. So this is kind of what the olfactory epithelium looks like. You've got these neuronal receptors, which are actually the neurons of cranial nerve 1. They're surrounded by these supporting or sustennacular cells and stem cells, and these are cells that support those. These are cells that support those and help those stem cells function and regrow and the nerves to work. And then there's the cilia on the surface, and that's where the olfactory receptors lie. Let's see what. Oh, I clicked save annotations and I. Okay. So olfactory receptor neurons, there's somewhere between 6 and 10 million. They form this very large surface area to detect odorants. The odorants bind to these receptors at the cell membrane and then activate this signaling cascade. And the way we smell is that we sniff into our nose. That creates turbulent airflow. The odorants then diffuse are transported through that turbulent airflow up to the mucus. They diffuse into the mucus, are transported in the olfactory receptor by chaperones called odorant binding proteins. And then these odorants bind receptors and then induce signaling. And these receptors were discovered by Richard Axel and Linda Buck in 1990. In the 1990s, in 1991, they discovered this family of olfactory receptor genes, which is an ancient and large gene family. And they discovered this physical encoding basis for the sense of smell. And of course, they were awarded the Nobel Prize in 2004. So to give you an idea of the timeline, physiology of hearing was discovered, say, in the 50s, and maybe physiology of vision was discovered in the 70s, and we're talking about the 90s for physiology of olfactory faction. So we're quite far behind in understanding how this works in humans. Interestingly, olfactory receptors are present on every chromosome, so they're an ancient part of our physiology. These genes have sort of infiltrated our entire genome, and people have shown that genetic variation in these olfactory receptors can influence olfactory function and perception. That's kind of a very interesting fact of this gene family. So, how about clinically? How do we think about olfactory system pathology? So, you can categorize it by cause as inflammatory or traumatic or congenital or degenerative or related to endocrine or neoplastic problems. You can think about it as location. So is this in the nose, is this in the nervous system, or is this systemic disease? And then the most common way of thinking about it is how is it working in the olfactory system by location. So Is this a conductive problem where odorants are blocked from getting up to the neuroepithelium? Is this a sensory neural problem where the nerves, the neurons are not working? Or is this central? Is this brain disease and it's a problem in the brain? In terms of common causes, the most common cause is aging, which is obviously our focus tonight. Post-viral smell loss has been reported, and until really recently, we really haven't identified specific pathogens, but now we know that SARS coronavirus 2 is a pathogen that causes olfactory loss, potentially via affecting those supportive cells around the stem cells in the olfactory epithelium. Inflammatory disease, which we all treat, rhinosinusitis or nasal polyposis, very common in younger people. And then trauma, so hitting your head, car accidents. These can shear the olfactory cilia as they go through the cribriform plate and injure the nerve. Now, there are other causes. Obviously, surgery to this area, either the nose or the brain, can cause this problem. Neurodegenerative diseases, which we'll get into in a little bit, and then other things like chemical injury, toxin exposure, endocrine diseases, such as Kalman syndrome, or tumors, which can affect the olfactory area or the olfactory nerve itself. So how do we measure olfaction? Well, many people are unaware of their olfactory deficits. You must perform objective testing. Now, there's a variety of different kinds of tests. There are fancy tests, there are simple tests. But this testing allows you to assess the degree of loss. It establishes the validity of disease, so you can rule out malingering. And then you can monitor changes over time. And this is particularly important in, for example, post-viral smell loss, which can recover, and you can monitor that recovery because people are not very good about self-reporting how good they smell. You have people who say, I can't smell very well, and they do great. You have people who say, I smell fine, and they don't do great. Part of that is because I think that people detect airflow and they think airflow is actually sense of smell, and that can be confused. There's a variety of methods, as I mentioned. Most commonly, you can measure odor identification. So you give someone an odor and you give them some choices and say, which odor is this? And they choose. So that involves not only detecting the odor, but also cognitively picking a choice and identifying in your memory what that odor is. You can either use the University of Pennsylvania smell identification test, which is a sniff and stick, a scratch and sniff smell test. You can also, in Europe, they use the sniff and sticks, which is a marker-based smell test, which accomplishes essentially the same thing, and they correlate reasonably well. There are cross-cultural versions of these tests. Some of them are validated and some of them aren't, but you get the same information. How well do people identify odors? Some more different and a little bit more advanced smell testing might be threshold testing, where you give people an odor. It doesn't matter what it is, but they detect at what concentration they can detect it. So it's a detection test, and obviously people who can detect low levels have good sense of smell, and those who require high concentrations have a poor sense of smell. And you can deliver this by upset, or there's some other ways of doing it. You can also test odor discrimination, which is here's two different odors and 3 different odors, and how are they different? Can you give them 3 choices? Which one is different? And lastly, you can do neurophysiology, so you can actually measure brain responses after presenting odors and see. And those latter 2 are more in the research realm, so not useful clinically. What the upset looks like, again, developed by Richard Doty at Penn. It's a 40-item scratch and sniff test. It's validated across the world, different populations. Its test retest reliability is over 90, over 0.9, and it's cheap, but it's not reusable, so it's like about $25 or so. There are age and sex norms available, so you can tell people how they do compared to their compatriots. And it's a standard test, easily available and easy to do. All right, so after you test people, after you see people go to hit, what are the important parts of the clinical history? So you want to know the onset, the rate of decline, and the duration. Slow decline would tell you something different than a rapid onset. Nasal symptoms are important, in particular, inflammatory. You're looking for inflammatory disease, so that's sort of the keys under the light. If you find it, you can treat it, and inflammation is something we can reverse. We have lots of treatments for that. You want to ask about facial or head trauma, prior surgeries, again, getting at that traumatic injuries. Neurologic symptoms are a little bit of a warning sign, which would send you towards things like neurologic disorders, neurodegenerative diseases, tumors that might present with neurologic symptoms. And then family history. Now, that's very rare. There are some syndromic diseases that are listed here that can cause it. There are also specific anosmias where people can't detect specific odors. And there's a few conditions where people have genetic mutations in the receptor signaling and they can't detect any odors. They're born without any sense of smell. So that's just something to think about. Medications can be associated with decreased sense of smell or impaired sense of smell. The big ones are cholesterol medications, psychiatric medications, and medicines that affect the brain because they can affect neuronal sensory function. You don't want to get a good history of a viral infection. Obviously, with COVID, people are very aware of that. In the past, people would have a cold, it would go away, their sense of smell wouldn't recover. They'd say, hey, it's going to come back, I'll just leave it alone. Months would go by, sometimes years. They'd see their doctors. People weren't as aware, and so it was hard to get good care. Now, I actually saw a patient, who was, about a week out from having a cold and came and saw me because they were worried about their sense of smell. So that's now in the consciousness of people to look out for with COVID. Occupational exposures, so people who are exposed to toxins, you definitely want to look into that carefully. Some of that can be inflammatory, but some of that can be neurotoxic. Diet. Now, you have to be very malnourished to have a poor sense of smell from your diet, from nutrient, micronutrient, or nutrient loss. So that's something you want to think about. Tobacco and alcohol have been associated with smell loss. And then, again, I mentioned drugs. I didn't mention illicit drugs, but certainly anything toxic that you put in your nose can injure your sense of smell. And intranasal zinc, which has been touted for its antiviral effects, actually injures the olfactory nerve and can be associated with olfactory loss. So you never want to give anybody that and and you could get a history of that. All right, physical examination. So you want to do a thorough nasal examination, including endoscopy. You're looking for inflammatory disease. You're looking for nasal tumors that you need to treat. We're good at that. Careful head and neck exam, complete neurologic exam, specifically cranial nerves, memory, any focal deficits. You definitely are going to push you towards getting input from your colleagues in neurology and doing more detailed imaging. And I don't have it here, but if you suspect inflammatory disease, you certainly can get CT scans of the sinuses. And then MRI hasn't been shown to be very effective. You can find things like smaller olfactory bulbs and some findings, but most of those are not actionable unless you find you're looking for specific neurologic diseases, MS, etc., or you suspect a tumor, in which case you can get MRI imaging. And then how do you treat this? So the best outcome, again, is inflammatory disease, conductive disease. So we're good at this: allergy immunotherapy, systemic or intranasal treatments, surgery as indicated, and that can be very fruitful. We're not as good at sensory neural dysfunction. In fact, our treatment is really to treat associated diseases, if it's autoimmune or diabetes or other things. Nutritional support, but again, most people are not deficient, probably doesn't hurt. And then olfactory training, where you expose people to odors, usually about 10 minutes in the morning and 10 minutes in the evening. There's 4 classic odors, although I don't think the odors make a difference too much, to stimulate either brain plasticity, smell growth, learning, can be shown to be helpful in a variety of settings of olfactory loss. And then counseling. So, one thing I find really interesting is, even though we don't have a lot of treatments for age-related olfactory loss, people are really happy if you can just explain some things about olfaction, explain some things about aging, and just tell them, like, yeah, what you're feeling is real, because they've gone to some other people, other doctors who, really don't know a lot about it. And you can be a nice expert about that and comfort them. You can also counsel them for things that are practical, like, warning them that they'll need help from family members to detect spoiled food. They need to throw things out by the expiration date. They need to install smoke detectors and monitor their cooking gas for leaks and change their batteries frequently. Be careful. And then I also emphasize to patients that they really need to work on other characteristics of foods, things like temperature and texture and visual appeal. They want to get pleasure out of foods in different ways other than flavor, which is going to be difficult with an impaired sense of smell. Right. That's sort of the background. That's sort of part one. I wanted to give you a sort of a good background of olfactory function, clinical workup and management, and just sort of a history of what we know about the olfactory system. I'm going to turn now to a couple of different projects we've been working on to understand how the sense of smell is related to aging and diseases that are important in aging. And the subject of my research in this area is the National Social Life Health and Aging Project. This is a longitudinal study of aging of 3,005 community residing older adults. They were ages 57 to 85 years old at baseline, which was in 2005. So this is like the greatest generation born between the years of 1927 and 1945. It's a probability sample representative of the United States population of older adults. So it's representative of all older adults in this age range who live at home. And we've used this group, this population, this project to study the interplay of social and medical issues, including things like elder abuse and social disconnectedness, sexuality in older adults, smoking and alcohol use, medication use. One of our most impactful papers, I think, was a paper in JAMA that showed, and this is not my work, it's my colleagues' work, just showing that many patients, older adults, if you just list their medications and you go through them, a huge percentage of older adults are on medications that are contraindicated together. So just really important to think about these things. Diabetes, frailty, sleep, and there's many other things. We have detailed and standardized health questionnaire data. These were people who were interviewed at home and measured at home, which is another little unique finding, part of the study. And then we measured vision, hearing, taste, touch, and smell at baseline. And then some of these measures were repeated in follow-up. So we have data collection in 2005, 2010, 2015, and we're currently collecting the fourth wave 15-year follow-up in these subjects. In the second wave in 2010, we collected data on the partners, the living partners of these people. And in 2015, we collected what's called the baby boomer cohort. So the cohort of older adults born during the baby boom who are now following over time to see differences in people who grew up in the greatest generation versus the baby boom. So the sense of smell was measured in NSHAP. It's a validated 5-item version of the sniff and sticks. And people were given a picture and a word for each choice of these five items, and then asked which one they chose. It took about 3 minutes to do it. And we categorized people in the olfactory function as either 0 to 1 correct, 2 to 3 correct was hyposmic, 4 to 5 correct was normosmic. So anosmic, hyposmic, or normosmic. 3 minutes, we could get this score. We also have things like race and ethnicity and cognitive function and physical health and comorbidities and mental health and smoking and problem drinking and socioeconomic status. We have a lot of things that we can adjust for that might affect health and aging in older adults. Our idea was, the sense of smell, here's like an anatomic cartoon of, odors come into the nose, they get signaled in the nose, the neurons project to the olfactory bulb, that projects to the olfactory tract, and then it's connected to parts of the brain like the orbitofrontal cortex, the thalamus, the entorhinal cortex, and the hippocampus involving memory. So, really close connections to important parts of the brain like pleasure and memory and emotion and decision-making. So really core parts of the brain. Because of that, it's been observed that olfactory deficits appear early in neurodegenerative diseases such as Alzheimer's. We have found, and many other people have found all over the world, different populations, different smell tests, different cognitive tests, you all find the same thing, which is that people who have decreased odor identification subsequently develop a dementia, indicating that olfaction is the canary in the coal mine for Alzheimer's disease. Now, all of these tests were odor identification tests, and as I mentioned to you, odor identification has a cognitive component, right? So you have to not only say, okay, this is lemon, You have to remember what a lemon smells like. You have to read lemon, and then you have to choose lemon, and then you have to say lemon. So there is a cognitive component to that. And maybe olfaction is just picking up the cognitive component. Maybe odor identification is just a cognitive test. Well, we can address whether it's actually detection of odors or identification odors by looking at threshold. And whether threshold, or just you can say at what threshold do you detect an odor? It doesn't matter if you know what it is or can say what it is. Does that is that linked to dementia? There's also little information about how genetics plays a role in the olfactory decline. So one genetic connection is the ApoE allele. So ApoE4 is a genetic variant that's associated with higher Alzheimer's risk and higher and earlier Alzheimer's onset. Those with the ApoE4 allele have worse odor identification, but there's limited data on odor threshold. Most studies of these are cross-sectional. They have small samples. They're homogeneous, or in Alzheimer's cohorts. So these are high-risk groups. And the question is this genetic link generalizable on a population scale? And does a similar link exist with odor threshold, which reflects more peripheral olfactory function? So you can think of odor identification as sort of peripheral and central olfactory function, and odor threshold as more peripheral olfactory function. So we tried to answer this by looking at the rate of decline of odor identification and odor threshold in ApoE4 carriers and non-carriers over 10 years of longitudinal follow-up in NSHAP. We tested this hypothesis that ApoE4 predicts central but not peripheral olfactory decline. So to do this, we looked at ApoE4 status. We had genetic testing in these patients, and we found that ApoE4 status is associated with olfaction and cognition in cross-section, as other people have found. So if you're in this high 4, E4 homozygote allele, you have worse cognition here. And similarly, these E4 carriers have some sense of smell problems where they have decreased threshold. We found that odor identification is indeed worse in ApoE4 carriers after age 75. So here you see the carriers in blue, and in black, and you see the non-carriers in blue, and you see at age 75 here, you start to see separation by genotype, where the carriers have worse odor identification scores compared to the non-carriers. So it's manifesting a little bit at about age 75 or later. And interestingly, we found that for threshold, this change in olfactory function occurs earlier. So here you see the E4 carriers again in black, and you see even as early as age 65, the carriers have worse thresholds compared to the non-carriers. It's sort of some overlap here, but you can see in general, the black bars are below these non-carrier bars seen in red, indicating that olfactory threshold occurs even earlier before odor identification manifests, odor identification problems manifest in older adults. And then we wanted to look at this question over time. So that's in cross-section. How about over time? And we find that ApoE4 carriers indeed have worse odor identification over time. So you see here are the E4 carriers at 5 years, and the non-carriers at 5 and 10 years here in green. And you see here at 5 and 10 years in red of E4 carriers, they have worse olfaction, lower odds of having good olfaction here, highly significant, indicating that people with E4 alleles have worse odor identification scores over time. Their sense of smell gets worse over time. And interestingly, E4 carriers have similar odor threshold over time. So here the E4 carriers are here. This is just time going by in non-carriers, and no difference in carriers in terms of odor threshold over time. We did find that known effect of ApoE4 on cognitive decline. So here you see people aging 5 years and non-carriers, and in carriers, they have worse cognition over time, indicating, what previous people have found, that people, when you have that ApoE4 genotype, you have a worse cognitive decline over time. So what we found here is that odor threshold deficits are apparent earlier at younger ages than odor identification deficits, and that and that ApoE carriers show worse odor identification over time, although threshold was similar, didn't show a change in olfaction over time. Cognition declined more rapidly in ApoE4 carriers as we expected. And so what that tells us is that peripheral olfactory function in ApoE4 carriers occurs before central olfactory dysfunction, consistent with prior work showing AD pathology in the olfactory system. And it seems like genetic influences appear to affect the rate of decline of central olfactory dysfunction, but not peripheral olfactory dysfunction. These data show us that olfaction can be connected with diminished cognitive function and ultimately neurodegenerative diseases, and that we might want to look at threshold as an even earlier manifestation of olfactory loss, even before you have odor identification deficits. And as far as that might be a better predictor of neurodegenerative disease. Now, I'll just show you another, I'll try to go through these last few slides quickly so we can have a little bit of discussion, but we want to look at the question of olfaction and depression. So I mentioned that olfaction is closely connected to mood. Cross-sectional studies have shown that people with olfactory dysfunction have more depressive symptoms. But the question is, which came first? Did you become depressed and that affected it affects the brain and that's what's causing your olfactory dysfunction? Or is it that you get olfactory dysfunction and because your quality of life is terrible, you develop dementia over time? And because of NSHAP's design, we could answer this question of how the sense of smell is connected to this part of brain function. And we ask the question, what is the direction here? So we, of course, defined olfactory dysfunction as more than two errors in odor identification. We have depressive symptoms measured by the Center for Epidemiological Studies Depression Index, or CESD. We had scores from 0 to 22. And we defined frequent depressive symptoms as scores of over 9. And then we built multivariate regression models looking for, did olfactory dysfunction predict depression over time and vice versa. And we can adjust for things like demographics and comorbid diseases and healthy alcohol use and smoking status and cognition, all the things that might be confounding this relationship. And indeed, we found that people with olfactory dysfunction had higher numbers of depressive symptoms. We found that people with depression had worse olfaction. Again, these cross-racial relationships, what came first, the chicken or the egg? And interestingly, we showed that the effect of olfactory dysfunction at baseline on the incidence of depression 5 years, 10 years later was really important. So people with olfactory dysfunction at baseline had 2.2 the odds of having depression, a new diagnosis of depression, increased depressive symptoms over time, 10 years later, controlling for all of these things. And interestingly enough, this really occurred in people who are healthy. So if you had no comorbidities at baseline, you had a very high probability of having, and had olfactory loss, you had a very high probability of having depressive symptoms or depression over time, whereas people who had at least one comorbidity, that relationship wasn't there, telling us that it's really in healthier people when you have an olfactory loss, it really is the big difference in your mental health. This is the first time that Simon has shown that olfactory dysfunction predicts the development of depression over time. Again, in people who had low comorbidity. I didn't show you the data, but depression did not predict the development of olfactory dysfunction the other direction, indicating that that's the directionality of that relationship. So this can disentangle the relationship between olfaction and mental health. It supports the use of olfaction as a depression screening test. And so when you find patients with olfactory loss, you really got to ask them about depression because it can affect their mental health and be a risk factor for their mental health. Useful clinically for that. All right, I'll just briefly end by talking about the ultimate health outcome, which is mortality. We showed that olfaction predicts mortality. People with olfactory dysfunction have 3 times the odds of death, adjusting for everything that you can think of that might be a risk factor for death. And in fact, when we looked at that comparison of anosmia as a predictor of mortality and compared to things like we all think are important, like heart attack and stroke and diabetes and heart failure, anosmia is a better predictor than almost all of these things short of heart failure. So really good predictor of mortality, especially compared to these big morbid diseases. But we wanted to know what about the trajectory of smell dysfunction, right? You get something in cross-section, you don't really know what the timeline is. And is that acquired dysfunction? Is it aging? And could we look at olfactory trajectory, the pattern of how olfactory changes over time? Is that a better predictor of mortality? So we looked at olfactory trajectory. So we have olfaction at baseline here on this chart on the left, and then on the top we have, oops, on the top we have olfactory status at 5-year follow-up. So either normosmic, hyposmic, or anosmic. So you can see here a big number of people were normosmic in both time points. Some were normosmic and turned hyposmic or anosmic. Some were hyposmic and their sense of smell got better, probably test error. And some were hyposmic and persisted as hyposmic or anosmic, etc. So we have these colors here: persistent normosmia, the return to normosmia, new hyposmia or anosmia, or persistent hyposmia or anosmia. And then, of course, we have, this is a longitudinal study. We know who died. We looked at who died at wave 3, so we could tell, the proportions who died. You can see here, just in the chart, that people who had persistent hyposmia or anosmia, 41% of those people died. And even people who had new hyposmia and anosmia, 20% of those people died. So those are pretty big numbers. But of course, lots of things could have been involved in those deaths. And so we looked at this, common model that we use when we adjust for age and gender, race, comorbidity, and cognition. And you can see here that this trajectory, these people with persistent hyposmia or anosmia still have a highly increased odds of dying over time. And that is not true for people who had either returned to normal sense of smell or had new hyposmia or anosmia. So it's really this trajectory of persistent hyposmia or anosmia that predicts mortality. And then we wanted to see, which was more important, just that kind of single time point analysis here where you see there is a trend towards predicting mortality, but again, more significant for those who remain hyposmic or anosmic, have that poor trajectory of smell loss. So it's really, this trajectory is even more important. And so you can ask people like, how's your sense of smell over time? Or you can test people over time, and it will give you a very good idea about what the risk is of 5-year mortality. So in conclusion, and I'm sorry that was a little bit of a whirlwind here, sustained olfactory decline over time appears to be a strong predictor of mortality than single time point assessment, consistent with the concept of sensory aging overall, reflecting overall health outcomes in older adults. I think of this as sensory frailty. So we know a lot about physical frailty, how important that is for surgical patients, how important that is for older adults. And I think of these kinds of things like hearing loss and vision loss and olfactory loss and taste loss and decreased proprioception and touch as analogous to physical frailty, and it has the same risks. This may be one way how you can identify high-risk older adults. A 3-minute olfactory test can give you a lot of information. So just to conclude, thinking about changes in sensory function over time can give you some clues about what changes in function might lie ahead, things like depression and dementia and death, which I've showed you today. There are other things. Conversely, risk factors such as genetics and demographic and clinical parameters can predict how sensory function ages over time. We should find ways of assessing patients about changes in symptoms over time and look ahead for more important sequelae, really identify people who need more intervention, and maybe identify people who need to be in clinical trials for dementia and mobility and cognitive interventions, social interventions. And then finally, changes in olfaction can predict major health outcomes. And that, I think, is something we all need to think about. So that's my talk. In conclusion, I'd like to thank the members of my research group and my colleagues who are working with me on this research, including colleagues at Boston University, Rush University, and Michigan State. I have funding from these bodies. In particular, I'd like to highlight the medical students who worked on these projects. Yazan Alayan did the work on depression. Matthew Goodsmith did the work on olfaction and genetics, and Dara Adams, our chief resident, who's going into rhinology, did the work on pollution and mortality and many other projects. So thanks very much, and if there's any questions, I'd be happy to take them.
Fantastic, Dr. Pinto. This was a wonderful presentation. Congratulations on your work and your accomplishments. The concept that you put forward of sensory frailty is as a unifying theme is pretty important. It was back in mid mid 2000s, early 2000s to mid 2000s, when Dr. Gates published his work on the ability of central auditory processing disorder tests being able to detect risk of Alzheimer's disease 7 years ahead of an actual diagnosis of dementia. And that concept has carried through to now people like Justin Galoob, who's here, and I'll unmute him so he can speak and ask his questions or comments. And yourself have demonstrated that this concept carries forward and is replicated, and not just hearing pathways, but also in olfaction. And so the question becomes, is it possible to use this as an index of predicting frailty and potentially in the perioperative stages and before the operation, if the person is identified, say, on a sniff stick test to have hyposmia or anosmia, is that patient potentially at higher risk of developing, say, delirium postoperatively? Are you aware of such a work or that kind of concept being around? You kind of hinted at that potential indirectly.
That's a great question. There's a lot of things to talk about there. I'd say, first of all, you see similar relationships for hearing, for vision, and for the sense of smell. So I think that tells us that there's a common theme here of sensory function and the brain being very symbiotic. Now, whether that's which came first, or whether the same mechanisms that break down those cranial nerves are also affecting the brain is unclear. Certainly, you can find Alzheimer's pathology in the olfactory system, and so maybe that's an indicator of the same pathology that's going on. And there are people working on olfactory tests as a screening method for even in primary care for risk of mild cognitive impairment and dementia. And there was also people doing work on restoration of hearing. So hearing aids so Frank Lin is doing some nice work on hearing aids to see if that could prevent the decline or mitigate the decline of cognitive dysfunction that's predicted by hearing loss. In terms of sort of inpatients, I'm not aware of people working on delirium specifically, but certainly you could use some quick screening tests in the hospital to predict some clinical outcomes. When we published our work on mortality, one of my colleagues said, you should just go to the dean and tell him you got to test every patient that comes into the hospital and then look at the outcomes. And, stupidly, I didn't take him up on that because I thought that was a little crazy. But then when I've done some subsequent work showing that, sense of smell can predict things like development of pneumonia, development of heart disease, really some other things that are important clinically. I think it could be used as a quick screening test. I'd love to hear Justin's comments about hearing. I know he's doing some amazing work on hearing and dementia as well.
Can you hear me?
Yes, we can hear you.
Oh, thanks. I just was able to share my video. Ignore my background. We had a little flood and there's no doors in my closet, so sorry about that. I don't want to steal your stage, but I sort of follow in the footsteps of Franklin and do similar work. But I've been fascinated by this, and one of my colleagues here, John Overdevest, also studies olfaction and its relationship to cognition, sort of the brain effects of olfaction. He just got a K award related to that. But yeah, thank you for speaking. I really enjoyed this. Is tremendous.
Yeah, I think these are all kind of common themes in our field that we can work on together. And we've done a little bit of work on looking at the senses in totality. So hearing, smell, touch, taste, and vision. We've showed that there's some physiologic overlap, at least in part, between the mechanism, something called global sensory impairment, and that kind of combined element of sensory loss predicts frailty, predicts some of these outcomes. And if we could kind of identify that common element, that I think that would be a really important way of helping our patients sort of mitigate sensory loss, across, many different modalities, which I think would make a big difference for older adults.
Yeah, I agree. Absolutely. Exciting
If
work.
I could ask one more question. And by the way, anybody else in the audience who wishes to ask questions, if you wouldn't mind, raise your hand and I will unmute you or put you on the panel so you can pose your question. But just another question that occurred to me was the strong association that you demonstrated, not association necessarily, but olfactory dysfunction being able to predict development of depression over time. It's a pretty powerful finding. And obviously, these results are in the context of the National Social Life, Health, and Aging Project. So we're dealing with an older population. Now, the prevalence of olfactory dysfunction has significantly increased because of COVID. So we also have a younger population that has similar deficits and dysfunction. So if they are followed over time, would you predict that their deficit will also lead to depression, or is what you're seeing really the effect of aging and the interaction of aging and olfactory dysfunction that results in depression in older life? So
That's a great question. Hasn't been addressed in longitudinal studies. Now we can do those because unfortunately we have all these COVID survivors who have smell loss. I would predict it will. If you just look in the popular media, like the other day I saw this article about this, 21-year-old college student got COVID, got smell loss, and, was giving this sort of fearful, really impactful TikTok video about how it was just affecting her life, and it was pretty moving, and it got a lot of attention online. There is, I guess what's different for older adults is sense of smell declines over time, very, very slowly over time. And, many older adults don't even recognize that they have decreased sense of smell. Whereas when it happens acutely, you're really going to notice it. And especially sort of in the prime of your life where your olfactory system's working great, take it for granted. When it's gone, you really feel the effects. So people who can't really taste flavor, just everything tastes the same. They taste like cardboard. And maybe that's not even the worst deficit. The worst deficit is, you taste something pleasurable and it tastes terrible, or it has a distorted flavor or odor, or you detect odors that aren't there. You have this thing parosmia where there's no odor there, but your sensory system is telling you there is, and you have this terrible odor all the time. So I predict there is going to be a huge impact of that, and really, really need better ways of dealing with post-viral smell loss to really kind of work on that. And it is going to be a big problem for us.
mm-hmm. Very interesting. Any other questions or comments? Excellent. Well, thank you again, Dr. Pinto, for a fantastic presentation. We wrapped up 2021 for us for the seminar series for American Society of Geriatric Otolaryngology and in an amazing fashion. We're very grateful, not only for your presentation, but your patience with me addressing the technical issues that we encountered tonight. So I apologize to you and everybody who participated. But just to reiterate, there is CME available for this presentation, thanks to Dr. Puskas and Duke Department of Otolaryngology. And in order to be able to get that credit, you need to set up a Duke OneLink account. And I sent the link earlier today. Once you have set that up, the code for the webinar was also included in the email that I forwarded you, DUZGEG. And all you have to do is simply text that code to the number provided, 919-213-8033, and you will be provided CME credits. So I want to thank Dr. Puskas for making this possible. That definitely helps. I just want to also let you know that we had 38 registrants for this from all over the globe, all over the United States, going from India through Middle East and to Europe. And I'm sorry that the technical problems presented a challenge for many because many dropped out. And that take the blame for that. But again, it's a credit to you, Dr. Pinto, for attracting such an amazing international audience. So thank you again, and I wish everybody a happy holiday season. Have a good night.
Thank you. Good night. Happy New Year.
Thank you.
Thank you.